Thank you, Janice. It's a pleasure to speak after 3 years of Zoom meetings for the first in-person meeting of the UCLA Health System Board and the 10th anniversary of the board as a board itself. So it's great to be here. And it's also nice to follow Michael Keaton and his touching description of stroke and how it affected his family and how it affects lots of individuals. And so this talk will be a really high-level, short talk about some of the devastating conditions in neurology and how we're bringing forward, through some of the scientists and clinicians at UCLA, new treatments and new ways to prevent the disease. Just as a show of hands, how many people themselves or with family have had a neurologic disease or been touched by that? So it's practically all the hands in the room, and the stats bear that out. And so with stroke, for example, in 2014 dollars, it's $110 billion a year. There's 7 million stroke survivors, and that incidence is expected to dramatically increase with the aging population. With Alzheimer's disease, there's 6 million people living with that disease, and in the next decade or decade and a half, that's supposed to double. There's 1 million patients living with Parkinson's disease, and that incidence is supposed to double by 2050. So it sounds sort of hopeless, a kind of a relentless progression as we age of neurologic disease. But what I want to present here is the future state of the brain and aging. Where is there hope? How are scientists and clinicians at UCLA capturing some of the principles of brain disease and brain aging and bringing them forward to the present so that we can treat some of these diseases? Lack of speed kills. So this is kind of taking a page out of Dr. Jeff Saver's book, who's a professor of neurology at UCLA, who has a kind of a famous quantitation of how stroke affects the brain and how millions of brain cells die a minute when that clot is stuck in the blood vessel, and miles of the axons, the connections in the brain, die per minute. And so speed is of the essence of stroke, and we've heard quite a bit about it, but the Arline and Henry Gluck Stroke Rescue Program was kind of a fun example to start off with. We've heard a bunch about it, but it really is an example of planning the future before it happens. And so with this gift propelling it, and Dr. May Noor working tirelessly to help here, and Janet Hahn really helping at the county level, they placed it into the actual EMS services, and that took tireless work in LA County and in some of the adjacent cities to get it active and routine just before major clinical studies came in Europe and the US that showed that putting a CT scanner and a neurologist in a stroke unit and driving it to the patient's door resulted in faster breakup of the blood clot in the brain and also better recovery. And so it's a future state brought to the present by kind of the foresight of Henry Gluck and others to get this going. A second example is also being led here at UCLA. A thrombectomy is when you take the clot out of the brain blood vessel. And we heard a little bit from Michael Keaton on how that is technical, really advanced, routine in UCLA Health, but not available in far-flung communities or in some underserved areas. And so what doctors like Dr. Satoshi Tadashima in radiology, May Noor, and others are doing is using robots, so a joystick drives at another site the removal of the clot. So we can extend this technology to underserved areas. And this is actually an ex-stroke fellow, Charles Beeman here at UCLA, driving this. Dr. Saver further went on to geomap where these need to go in California in order to really reach underserved communities. Brain plasticity in a pill. So that's a future state that is some of my work. And it's really an interesting and unique UCLA story. So I collaborate with one of the leading learning and memory researchers in the world, Dr. Alcino Silva here at UCLA. And Dr. Silva wanted to do this crazy experiment. He said, we need to identify new molecules to enhance memory. So he got all of the smart mice, all of the mice with one mutation in their genes that were smarter. He brought them all to UCLA. He retested them. And he found, to great surprise, an immune molecule had the greatest effect at enhancing memory. We were already collaborating, so he said, Tom, let's try this in stroke recovery. And sure enough, it had a dramatic effect on enhancing recovery in experimental models of stroke. And then we found a group of people in the world that are born with a mutation in this gene. They're like the genetic model for the mouse. And these people actually also recovered better in stroke. And so this paper came out in the journal Cell, but even before the paper came out, we were in international clinical trial. So it was bench to bedside even before publication. And that was a unique UCLA collaboration. This is work of Dr. Dan Geschwin and others at UCLA, and they're identifying drugs that stimulate regrowth of injured nerve cells in spinal cord injury and peripheral nerve in particular. And this uses deep data analysis and omics studies. It's a unique UCLA collaboration, and it captures a future state of the brain and aging in helping to understand how we can promote regrowth of the brain. Aging is malleable. So we think of aging as this relentless, hopeless progression of degeneration and decline. But a future state of the brain and aging is that aging is malleable. We can identify how people are aging. And indeed, we found that not everyone ages the same. Of course, we would guess that. But importantly, not everyone's brain ages the same. And so some of the work at UCLA is to understand indicators or biomarkers so we can understand how people's brains are aging and how they're experiencing or whether they're at risk for cognitive decline. This is the work of Dr. Noah Zeitlin, Tim Chang, and Jason Hinman and others in UCLA Neurology and other departments. And they're using the deep data of the electronic health record, or EHR. We all check into that, and our clinicians check into that. This is this huge collection of data that we can understand who's progressing, for example, in aging, who is experiencing cognitive decline. And then the Institute of Precision Health has done such a great job with genetics, we can say, what are their genetics? Are there genes that might be a risk for that cognitive decline? And then to highlight one element of work, Dr. Hinman has found a biomarker or an indicator in the blood of brain blood vessel inflammation. So a group of patients that aren't experiencing symptoms yet, but they may be at risk for a disease called vascular dementia. So a future state of the brain and aging is to draw a blood biomarker and find, perhaps unexpectedly, that a patient has brain blood vessel inflammation, and we can treat that. And this is a targeted aspect tailored to the aging brain based on these therapies. Breaking Parkinson's disease. So Parkinson's disease is this maze of interconnected molecules and symptoms, the way the brain cells die. It's been hard to break through this to understand and develop a cure. In research funded by the Laurie and Stephen C. Gordon programs in Parkinson's disease, this is really propelling work that allows us to move towards a cure in Parkinson's disease. Stephen Gordon calls it revenge research because of his father's illness. And we're asking three questions here. What if we could identify the different diseases that are together seen as Parkinson's disease? It's likely several different diseases that give the final endpoint. What if we could determine their risk and prevent them? If it still occurs, how do we track and stop Parkinson's disease? These top two is the work of Drs. Jeff Bronstein, Beata Ritz, Tim Chang, Kim Park, and others. And they're using some really interesting ways to understand risk for Parkinson's disease. And what they've developed is what's called a polygenic risk score. So we can use genetics to look across our 800,000 patients at UCLA and with a little more work identify who's at risk for Parkinson's disease just based on their genetics. The future state is to take this even further because we've identified there's only a handful of genes that give Parkinson's disease, that produce it. But there are hundreds of genes that are risk genes that may make it progress faster. And these risk genes have been really hard to find because they only come out when there's an environmental exposure at the same time. It's kind of a two-hit thing. You have a genetic risk gene, and then you might have a pesticide exposure or have airborne particle exposure, or it might simply be age that's the second hit. How do you find those genes that put a patient at risk so you could modify that second hit? And so Dr. Tim Chang, Kim Park, and others, Noah Zeitlin, are using the deep DNA data from UCLA Institute of Precision Health, plus that electronic health record data to figure out what are those genes and could they lead to new therapies. A final element is when Parkinson's disease still occurs, these abnormal proteins in the brain called alpha-synuclein circulate. It's almost like an infection. They progress through the brain. Doctors Carlos Patera Cayal, Will Zeiger, and Chao Pang are mapping this and understanding how this progresses through the brain so that it can be tracked and blocked. And Dr. Chow Peng is collaborating with the California Nanosystem Institute, which you may have heard of, which is allowing massively parallel screening of new drugs to stop that progression. So a future state in Parkinson's disease is to prevent it by predicting who will get it, but then to give to give new drugs that will block the progression for those that do break through that prediction. Precision medicine neurology, this is a sophisticated group, so I don't need to go through precision medicine, but I'll go through a little bit of how it's impacting neurologic diseases and leading us to a future state. This work really centers around a novel program we created in UCLA Neurology, first and still only of its kind in the nation, and this is the Clinical Neurogenomics Research Center. This is led by Dr. Brent Fogel. It allows us to routinely gather the genetic data with consent from all our patients so that we can predict disease onset for them, so that we can understand disease risk, and importantly, so we can carry that forward to develop new therapies. And these are some of the areas in which this is being applied. Precision medicine in Alzheimer's disease leading to hope, precision medicine and brain repair leading to regeneration, and precision medicine and brain stimulation leading to recovery. Personalized brain stimulation, this is the work of Dr. Kevin Bickart, a new faculty member in UCLA Neurology. So head injury affects so many people with concussion, multiple concussions in sports, car accidents, military blast. Most people recover, but the patients that don't are very heterogeneous. Some have trouble with learning and memory, some have trouble with emotional regulation. You can't give them a drug because that would treat all of them. They're so different. You have to personalize the therapy. And what Dr. Bickart realized is he could map the brain using functional MRI and look at all the connected circuits in the normal person and understand what's abnormal in the patients that are in particular having trouble regulating their emotions. And so fear avoidance occurs commonly after head injury and is like a force amplifier. It amplifies PTSD, depression, anxiety, and even memory dysfunction. And what Dr. Bickart found was surprising. He found a hyperconnection, an actual increased connection between the amygdala and the frontal lobe in those patients that were having trouble with emotional regulation. That's the precision medicine part. He could identify among the large sample size who was having this abnormal connection. And now he's developing transcranial magnetic stimulation actually right through the frontal lobe here to decrease the strength of that connection, and has a major clinical trial to test whether that's going to work. So a future state in traumatic brain injury is personalized brain mapping and then personalized brain modulation to enhance recovery. Precision medicine in Alzheimer's disease, this is what we know. There is no therapy at the moment that modifies this relentless progression from the normal brain to early memory or emotional dysfunction to the demented condition. Using precision medicine and other approaches, there are three main areas of interest and work here at UCLA. The first is the recently created UCLA Alice C. Rubin Integrated Memory Clinic. And this was from a far-sighted gift of Evan Thompson with Connie Frank. And what this will do is make sure that we integrate through the electronic health record all the patients coming in with dementia complaints or memory dysfunction. Right now, they might go to primary care or neurology or geriatric psychiatry or geriatrics. What we want to do is link them all together, understand what the diagnosis was, what the genetic testing is, what does the brain imaging show us, and as these new therapies come down the pike, how are we delivering them, how are we monitoring them, so we integrate the care so the patients have a common door, even though dementia is seen so far afield. A second major area of focus is in the UCLA Mary S. Easton Center for Alzheimer's Disease Research. This is led by Dr. Keith Vossel, who joined us recently at UCLA and has really taken charge of this to develop, in one example of all this work, I'll cite specific treatments. And so just like Parkinson's disease, there are subtypes of Alzheimer's disease. We see the end product, but there are many different ways in which it progresses. And one of the key discoveries from Dr. Vossel is that some Alzheimer's disease patients have subclinical seizures. We didn't expect this. They're not the big motor seizures that we see. They often occur at night. And Dr. Vossel was able to determine which patients had these and then treat them with just an anti-seizure medicine. not a big hullabaloo of a new clinical trial. And he's recently shown in an NIH-funded trial that that successfully reduces disease progression. And so that's an example of a specific treatment coming out with a subset of Alzheimer's disease therapies. We have under this umbrella the Seidel-Kagan Research Clinical Trial Unit, which Dr. Vossel runs, which is really providing cutting-edge clinical trials to our patients as well. The final element is a program just rolling out, and this is the UCLA Collins Precision Medicine of Alzheimer's Disease. This is led by Dr. Tim Chang. And just like Parkinson's disease, it's using the vast electronic health data to develop phenotypes. What do patients look like? What medicines are they on? What zip codes do they live in? How close to freeways are they? To agglomerate all this data of how people live, pair that with their genetics, and so we can identify genes that themselves don't stand out. Just like Parkinson's disease, there's only 4 or 5 causative genes for Alzheimer's, and we've nailed those. But there's hundreds of genes that convey increased risk of progression. And interestingly, some of them code for resilience. Some Alzheimer's patients don't progress. We'd really like to know what's giving us brain resilience. How about if we get a drug to treat that? And so that's a goal of the Collins Precision Medicine of Alzheimer's Disease Therapy. So we can do this. We can take that progression and prevent it or reverse it. I have just a short talk, so just a few more. This is the last Precision Medicine and Brain Repair title. This is the work of Dr. Stephen Kramer. So precision medicine in recovery, in brain repair. Dr. Kramer followed a theme that's common to the precision medicine field. If you take all stroke patients and try to test the drug on them, they're very variable in genetics and where the stroke occurred. You're likely going to fail. People are so individual. And so Dr. Kramer's new trial with a drug that enhances dopamine uses genetic testing to identify patients that are going to respond to that dopamine therapy, that are dopamine responders. So his trial has as an entry point genetic testing to get the patients that are going to be predicted to respond to the drug, so you just don't give it to all stroke patients. He's further understanding which lesion, where the stroke occurs in the brain, and those that are likely to respond to it the most. So this concept is personalized neural repair therapies in stroke, and a future state of the brain and aging in this field is in precision medicine, so the therapy is targeted to the kind of stroke and the genetics of the stroke patient. Second to last concept here, and that is stem cells in neurology. This is certainly a buzzword, and we've seen this in the lay press and in many different areas, stem cells and their effect on regenerative medicine. In neurology, there are 2 main areas in which stem cells have started to have significant action. This happens to be work in my lab, and this is, again, another unique UCLA story. So UCLA is the UC with the most people on the smallest landmass. We're all over each other. And this really works to the benefit of clinician scientists because we're next door to each other. And so I was next door to a symposium put on by a stem cell biologist named Bill Lowry. And after his seminar, I went up to Bill and I said, you know, that stem cell finding, he had a finding in stem cells in a dish, is likely to lead to something that might work in a kind of dementia. And so fast forward 6 to 7 years and many papers, we've now found and developed and patented and have companies that are interested in moving this forward, a stem cell that turns into a kind of cell that helps the connecting areas of the brain, the so-called cerebral white matter, repair and form new connections. And so that's an example of stem cell biology and how it will help the brain. And then this is work of Dan Geschwin, Jessica Rexach, and other neurologists to look at how you might take stem cells that are human, make them into a mini brain, a so-called organoid in the dish, and then you're not doing an animal model, you have human brain connection models. And they're starting to use this to understand how inflammation leads initially to brain repair in dementia. We think about inflammation as universally bad. But what Dr. Rexach has found that inflammation is initially good, and then it starts to amplify the disease. And she's identifying molecular targets to stop that using stem cell technology. So a future state of the brain and aging is stem cells as models for disease to design new drugs and as transplanted repair elements to help repair the brain. This is my concluding concept, and it's string theory of the brain. And many will have heard of string theory, but I put the definition up here. a unified description of gravity and particle physics, a candidate for the theory of everything, and a self-contained mathematical model that describes all fundamental forces of matter. So string theory of the brain is a unified understanding of concepts present in all brain diseases. This is a future state of the brain and aging. If we can identify in one disease, epilepsy, how to repair the brain or how to prevent cells from dying, and we're really good at this, it might have the same consequences in dementia or in stroke or traumatic brain injury. So one drug might be applicable across brain diseases. And that's in part what I mean by a string theory of the brain. And so to get back to Michael Keaton.
Hey, what are you doing? Hey, stop it. Hey, you're messing up here. Come on. Whoa, whoa, stop it. Whoa.
So current neurology is advanced, but it's still limited by a small view of the brain. And so how can we develop an expanded view of the brain? And so I could talk a lot about this because it's a great topic. I'm, in the interest of time, just going to highlight 3 areas. So one aspect of string theory of the brain is in the brain disconnectome. The brain itself is a so-called connectome. It's a complicated and intricate series of brain areas that connect to each other and fire in precision that gives us the poetry and pirouettes that distinguish us as human. And neurologic diseases produce disconnection: stroke, traumatic brain injury, epilepsy, dementia. Understanding how the brain is disconnected will unite our understanding across brain diseases and allow us to identify therapies that might work in many different brain diseases. And so that's a key aspect in the future state and a string theory of the brain. A second is stem cell aging. This is work principally by Dr. Tom Rando, who's a professor of neurology and directs the UCLA Broad Stem Cell Center. And so every organ has adult stem cells in it, including the brain. And these act to sense stroke or sense disease and repair it. And what Tom Rando has found is these stem cells age abnormally. They experience what's called genotoxic stress. Their genome experiences mutations and stress. They just don't handle it as they age. And so a future state of the brain and aging is to understand how we can get our stem cells rejuvenated, how we can take those stem cells through genetic modifications and make them more juvenile so they'll repair and replenish the brain. And the final concept is brain repair across all neurologic diseases. We tend to think of the brain as the diseases as the injury, the stroke, the head trauma. But there's a limited but partially effective phase of repair across all brain diseases, even in the early stages of Alzheimer's disease. We're starting to identify the mechanisms of repair that, even though limited, are like strings uniting through all the rest of the brain diseases and might allow us to have common therapies across them. So the key issue here is to return to the uniqueness of UCLA. These are physicians and scientists driven by abject human suffering that we see in our patients, lack of effective therapies, and a drive to go forward to take the bench to the bedside and the bedside to the bench so that we can use both sides of that equation to develop new therapies. And the future state will be in many of these coming soon in the next really 5 to 10 years.